Hello, everyone. It's great to be here, and I really enjoyed the previous presentation. Even though I knew some of it, it was nice to see it in history, and like historically and in context. So I'm going to tell you something a little bit different, as Kaya talked about, which is not a seq technology, but talking about how you perturb gene expression in chromatin. So as Kaya said, my name is Lacra Bintu. I'm an assistant professor at Stanford in the bioengineering department, and my lab is interested in both understanding the basic biology of gene regulation and chromatin regulations in human cells primarily, but also developing tools for controlling it. And we think that being able to directly control gene expression in chromatin state is essential for understanding a lot of basic biology. A lot of processes depend on gene regulation in chromatin, from development to immune response to our brain response to aging. And if we can really understand the basics of it, the hope is that we can develop health applications from reprogramming cells to really understanding and curing certain diseases that have to do with these processes. And what I'm going to focus on today is how we could do that by kind of specifically targeting particular loci or genes in the genome to activate or repress them. And I'm going to talk a little bit about CRISPR-I and CRISPR-A, so CRISPR interference and activation technologies. And I'll explain kind of what that is. But before we jump into how we perturb chromatin, let's talk a little bit about the basics of transcriptional control. So let's start from the bottom here. So in prokaryotes, which is not the topic of this talk, as I told you, I'm very focused on eukaryotes and specific humans. But in prokaryotes is much simpler. So activators, generally speaking, bind near a promoter that drives a gene to make the DNA into RNA and into protein. And the activators usually directly interact with the RNA polymerase that reads that gene. So they they help recruit the polymerase there. That also happens in human cells where the situation is more complex because you are in the context of chromatin. So the DNA is shown here in black, and nucleosomes are these balls that compact the DNA, but also serve as regulatory knobs. So there, if the chromatin is kind of open and the site where the transcription factor can bind is free, the transcription factor can also bind there and recruit, either polymerase directly or like general transcriptional machinery. It's a bit more complicated in eukaryotes. However, sometimes that locus is not open, is actually closed, and there are transcription factors that just that actually act on chromatin to create a permissive or active or accessible chromatin region, so that the ones that recruit polymerase and polymerase that itself can come there. And then kind of the opposite of that are repressors. So in prokaryotes is kind of more simple. Generally, repressors just compete for binding with RNA polymerase, but in general, that's not the case in eukaryotes. So most often in eukaryotes, repressors actually act on chromatin to put modifications on either the histone tails or on DNA that lead through recruitment of many complexes that I'm not showing here to a repressive chromatin state that is generally compacted and maybe at a place in the cell where it kind of precludes access to activators and transcriptional machinery to that gene. So this is kind of these are the general principles. And the nice thing about these transcription factors in eukaryotes is that they're somewhat modular for the most part. So if you look at one of these transcription factors that is shown here, they can generally be described as having a DNA binding domain. And an example is shown here, like, for example, zinc fingers are a particular DNA binding domain that recognize a certain DNA sequence, and in fact, they can be programmed to recognize particular DNA sequences. And then they generally have separate effector domains. And what are these effector domains? I'm going to talk about them a lot. So those are the ones that are the activating or repressive effector domains that actually most often are just small domains that interact with bigger complexes that are called co-activators or co-repressors. And so those could be either the transcriptional machinery mediator, which is part of, again, activating transcription, were chromatin regulators that change the chromatin state to make it active, such as histone acetylases. Now, if this effector domain is a repressive effector domain, then it would recruit co-repressors that most often are acting at the level of chromatin. So they are going to remove, their deacetylases remove active modifications or put repressive modifications, such as certain type of histone methylation or DNA methylation. And because they're generally modular like this, it actually gives us a great opportunity to reprogram them and control gene expression. So actually in the last 10 years or so, there's been a lot of excitement about controlling chromatin, because we think it has a tremendous potential for how we do mammalian synthetic biology and health applications. And what is shown here in this picture that I took from this review is the idea of having a programmable DNA binding domain, meaning a DNA binding domain that you choose where to target it in the genome, fused to one of these effector domains that could either drive transcription or change the chromatin state at that locus. And the hope is that if you manage to get this to work reliably and predictably, you can do a lot of things, like you can reprogram cell types. You actually understand better, because we don't actually quite fully understand epigenetic regulation that well yet in human cells. You can run screens that sort of connect the genotype with the phenotype. And the idea would be that eventually you can kind of develop novel gene therapies. So what could these programmable DNA binding domains be? So I alluded a little bit, initially they were zinc fingers, but that is hard, because there you have to design completely new proteins for each target DNA that you want to address, to sort of regulate. And the excitement in 2016 was right after the CRISPR system was developed. So the CRISPR system is a slightly easier programmable DNA binding domain. So I'm showing here a picture from a review from one of my colleagues, Stanley Chi. And initially, when the CRISPR system was discovered, it's this enzyme, one of them, there are many types of CRISPRs now discovered, but one of the original ones was Cas9, that when bound to a particular RNA, can use that RNA by base pairing with the DNA sequence to sort of bind specifically to whatever sequence you want to target in the genome. And the original enzyme actually cleaves, it cuts the DNA, and you can do a lot of cool stuff with that. But interestingly, if you actually do a couple of mutations near the active site of the CRISPR enzyme, of the Cas9 enzyme, you can prevent it from cutting, but still allow it to bind in the same manner where you can program what it binds. You can tell it where to go by the RNA sequence. And this now can become a DNA binding domain that binds wherever you want it, depending on what guide RNA you give it. And interestingly, in bacteria, because that's how transcriptional repression works in bacteria, actually just the binding of the deactivated, catalytically inactive Cas9 can actually block transcription. But that is not quite the case in eukaryotes. Roadblocks can work a little bit, but not that well. So this is from an older paper where again, my colleague Stanley Chi and people in Wendell Lim, Adam Arkin, Jonathan Westman, Jennifer Donald's lab had targeted a reporter gene that expresses a fluorescent protein, so you can see what's going on with it, and had shown that actually if you just put the Cas9 there, you can reduce expression a little bit, but it doesn't work. It doesn't go completely to kind of background levels. And why might be the case? It's because in general, eukaryotes, they have nucleosomes, so they kind of learned how to get stuff out of the way of transcription. And also in eukaryotes, most often you need to manipulate the chromatin state to change transcription. So actually what they found, from the same groups, what they found that works is actually fusing Cas9 that you now can send somewhere, wherever, with these effector domains from transcription factors. So either with the transcription repressive domain or the transcription activator domain. And In particular, what they showed in this paper from 2013 is that if you fuse DCAS9 with this crab from zinc finger 10, you can actually reduce gene expression much more. So it's kind of shown here a lot more than if you just put DCAS9 alone. And so who is crab and what does it do? So crab, as I said, is a repressive domain. It happens to be from a transcription factor called zinc finger 10. There are actually almost 400 crab domains in human proteins, and I'll come back to them. But in general, and this crab domain from zinc finger 10 was actually known since the 1990s because people had discovered it and had used it in synthetic biology, fused to DNA binding domains from bacteria or yeast, such as TETR or GAL4. So this had been done, but not in the context of kind of the DCAS9 programmable DNA binding domain. But how does crab work? So in its natural context, the zinc finger 10 has obviously zinc fingers, and then it has actually multiple crab domains that can bind this adapter protein called CAP1 that serves as a hub to pretty much recruit things that remove positive histone modifications, such as acetylation, and write kind of repressive histone modifications, such as histone-free lysine 9 trimethylation, and recruit also the reader of this particular repressive histone methylation that compacts chromatin called heterochromatin protein 1. So practically recruiting crab, either in its natural context through zinc finger 10, or by fusing just the crab domain to deactivate Cas9, creates heterochromatin at the locus. And that has been shown by multiple groups at this point. And that's why it works so well as a repressor. But we were wondering sort of like, how about other repressive or activating domains? Like, can you kind of, do they all work the same? What dynamics do they have? And because this process usually puts histone modifications, can you also impart epigenetic memory? So in particular, if you recruit a repressor at a gene, how much do you change gene expression? How fast? How does it happen in single cells? Actually, this is something Kaya alluded to. And then if you remove the chromatin, the repressor from there, the modifications most likely stick around. How long does the change in gene expression last? So I actually started working on this as a postdoc before the CRISPR craziness started. So we actually used a DNA binding domain that's borrowed from bacteria, which is called reverse TETR, that we can recruit to a fluorescent reporter that is site-specifically integrated. And when I started, it was in a human artificial chromosome. More recently, we introduced it in human cells at a safe harbor locus, which is a locus that is kind of active and doesn't get silenced by cells, and you can integrate it there without messing up too much stuff. And so we express in cells that already have this gene that expresses a fluorescent protein, we express this fusion between a DNA binding domain and whatever transcription factor or chromatin regulator effector domain that we want to study. And by adding doxycycline to the cells, we can induce this DNA binding domain and the effector domain to come to the gene, presumably turn it off. We started with repressors. And by washing out doxycycline from the media, we can release it and look at the epigenetic memory. And here's how the data looks like. This is published actually a bit ago. So when we recruit a particular chromatin regulator to the reporter gene, and we started with a chromatin regulator that's associated with histone methylation, a particular type, histone-free lysine 27 trimethylation. It's polycomb. It's less important exactly what this is, but it's a particular pathway of histone methylation that was known to be associated with heterochromatin. What we see when we follow individual cells by microscopy is that cells start on, this is the citron color, this is just the control that's supposed to stay on. The individual cells flip from on to completely off, but they do so in a population at different times. So this is one cell lineage, this is an identical, sort of a sister cell, but kind of does it at a different time. So in effect, what recruitment of this chromatin regulator does, it controls the fraction of cells that are off as a function of time. And actually, when we wash doxycycline and release the chromatin regulator, what we see is the opposite. Cells pop back on suddenly, And again, it's a process that actually takes much longer in this case. It can take days. We can only follow them, for a few days by time lapse, and then we switch to sort of flow cytometry, where you can measure the distribution of the reporter fluorescence, and you can see some cells are completely on, and some, even after 7 days after we remove doxycycline or release the chromatin regulator, some are still completely off. So it's very bimodal. And it can last, like at the level of the population, some cells remember and stay off for as long as 30 days. And we were wondering, okay, now how do different kind of pathways work, effector domains or chromatin regulators associated to different pathways work? And in that initial paper, we only tried 4. We tried to choose from different pathways. So we tested the CRAB one that I just told you. It's associated with histone methylation, and indeed is one of the best ones. It's very fast. And in this context, when we recruit it for 5 days, it has some amount of memory. Half the cells remember that they've been silenced. It can vary depending on how long you recruit it and at what locus. Things like DNA methylation seem to work slower. However, they have much longer memory. And things like histone deacetylation, they also work fast, but when you remove it, none of the cells remember they've been silenced. And at about the same time when we published this paper, actually, we're very happy to see that other people have seen similar things where CRAB acts fast, but so here CRAB is in red, acts fast, but has limited memory. Another DNA methylase is very slow to silence, again, as the percentage of cells that you turn off expression, but has memory. But then you can combine them. They actually combined them, and they said, okay, yeah, like if you combine CRAB with the DNA methylase, you can get now from just a transient kind of targeting of the gene, something that acts fast and has epigenetic memory. And people are very excited about that. So this was the paper in 2016. And actually, there's very nice work that came out from James Nunes from Jonathan Weissman lab just last year, kind of sort of following on this paper and optimizing what they call a CRISPR-off, which is this fusion between DCAS9 CRAB and DNA methylase. But even in the original paper, I wanted to sort of emphasize that it's not always true. So this is shown in a particular cell type, but even when they look at exactly the same reporter, but in a different cell type, they actually don't get such good memory. And we still don't understand that as a field. So this is still an open question. But there are other issues with kind of fusing this full-length chromatin regulators to cells to do this perturbation. So while CRAB is relatively small, actually, because it's borrowed from a transcription factor that itself is miniaturized, the DNA methylases that you need to get to this type of memory, actually, you need 2 of them, the main one and a helper one, and they're quite big. And then the whole tool becomes hard to deliver. You can't fit it into viral vectors that you would want to use for therapy like AAV. And and sometimes actually, which I'm not saying here, sometimes overexpression of this DNA methylases is actually toxic to cells. So we thought of 2 possible approaches. We and others in the field actually thought of possible approaches to kind of get more compact regulators that recruit the endogenous complexes instead of having to do this kind of Frankenstein fusions. And one that I'm not going to talk about is actually to develop nanobodies that get endogenous chromatin regulators. And the other one that I am going to talk a bit more is to use the natural effector domains from the human transcription factors to recruit in the same manner as the transcription factors do, just now where we can retarget it to whatever gene we want. And that's the one I'm going to say a little bit about. So how do we?
5 minutes left, just letting you know.
Thank you. Yeah. So how do we find better and more compact transcriptional effector domains? So I'm going to talk about this method that we've developed recently that's published actually in 2020, where we use the pooled method to test about 10,000 protein domains in one pot for their effect on gene expression in human cells. And this was developed by a graduate student, Josh Tycho, who is joined between my lab and Mike Basik's lab. And I wanted to say before I tell you what we did is that actually the entire field is moving towards using high-throughput approaches to characterize activators and repressors. And I've kind of listed here, it started with yeast, Drosophila, but now of recent time, even since we published our paper, more papers came out in human using kind of high-throughput approaches. They're beautiful work that I urge you to take a look at if you're interested in this topic. So here's how our workflow looks like that allows us to measure 10,000 repressors and activators. So we start with this fluorescent reporter that's integrated site-specifically in human cells at the AVS1 locus. And then we order from Twist actually a library of 10,000 DNA sequences, 10,000 oligos that come in one single tube. And we PCR amplify them and we clone them as a pool in one tube into lentiviral vectors, and then we make those into lentiviruses and infect cells at very low infection rates, such that now each cell gets a single effector domain fused to our DNA binding domain that can bind to our reporter. So practically now every cell becomes a test tube that has a single domain that can be recruited to our reporter. And if that, in this case, if that domain happens to be a repressive domain, and it turns off our reporter, we will see that the fluorescence will actually turn off. And in fact, we also added a surface marker that is expressed on the surface of the cells to allow us to do quick magnetic separation of the cells with an antibody and magnetic beads. So we can very easily separate the cells that have turned off gene expression from the ones that stay on. And then we say, okay, let's look at what domains do we find more often in the population that turned off. And the way we do that is actually by counting by sequencing, by next generation sequencing. So we extract genomic DNA from both the off and the on population. And then we sequence just the DNA sequence that's now encoding for the domains. Then we count how many times do we find a particular domain in the off population and how many times do we find it in the on population. And repressors are going to be found in the off population a lot more often than in the on population. So we can calculate very quantitatively an off to on score that will tell us whether domains are repressors or not, and the opposite is true for activators. So we did this. We measured now silencing for all domains that are already annotated in PFAM from nuclear proteins that are smaller than 8 amino acids. That's our limit of synthesis. And actually, what we found, so here I'm showing the off to on, like repressors are in this corner. Actually, a lot of them are these CRAB domains. And the very interesting, and actually they're the ones who have better epigenetic memory as well. And just one little tidbit that was super interesting, actually the CRAB domains have evolved in an arms race with transposons to sort of silence them through vertebrate evolution. And what we found is that the most recent CRAB domains are actually the ones that silence, while the really old CRAB domains do not silence in our assay anymore. And in fact, when we looked at activation instead of repression, we found that one of the really old CRAB domains was one of the strongest activators in our space. So activators are in the other corner because they have low off to on. And in fact, we used this new CRAB that was not known before to be an activator, but we found to be a very good activator to incorporate it into a new CRISPR activation tool. So let me just show you that part. So we attached the new kind of activator domains that we sort of found in our screen. We combined them, zinc finger, FOXO, and COA, into a triplet that's very short and actually can activate quite well gene expression at that endogenous gene, similar to the previous CRISPR activator, which is DCAS9 VPR, which is longer and fused to viral activators. And we think that this actually is a much better tool because when we deliver it to cell, what we see is that they both activate, which is shown here in the fluorescence of the gene that we're targeting, the CD2 on the x-axis, but the new tool that's this free component human activators ones has much higher delivery of the DCAS9, while we find in our hands DCAS9 VPR, we never can deliver very well. It seems like it's kind of toxic to cells and they kind of reject it. So we're very excited about this. This is unpublished. But what I wanted to emphasize is that, we have this method now to measure, we measure transcriptional activation and repression for many protein sequences within live human cells. And we used it to study families of domains in unpublished work to annotate repressive and activation domains and to build better DCAS9 activators or DCAS9 repressors. And there's a lot more work to do to test it at different genomic loci and different cell types. But this is a method that you can use to test protein function for whatever your favorite process is, as long as you have a fluorescence reporter. And with that, I want to thank my amazing lab, especially Josh Tyko, who was one of my first 3 students and just graduated and is moving on to his postdoc now. And to my collaborator, Mike Basic, who really helped bring the high-throughput methods into my lab that he was using before for CRISPR screens. And thank you all.